So thanks to all of you who have turned up today. I know it's been a very, very busy, full-on week with the Food Security Summit, but perhaps this will be a refreshing end to the week where we won't hear over and over again the words sustainability and food security. So we're continuing the theme of ancient DNA. And while we covered plant domestication in the previous seminar by Hernan Burbano, we'll be looking at an example of animal domestication today, namely the horse, which is an animal that's played a very important part in human civilization and history. So in this capacity, it's really a great pleasure to introduce Orlando Ludovic. Orlando, he graduated from the University of Lyon in France in 2003. He was appointed as an associate professor in 2005. And in 2010, he moved to the Center of Geogenetics in the University of Copenhagen, where he was appointed a full professor in, I think that was you were with that appointment, right? You were made a full professor, yeah. And I have just tested Orlando, and he does understand a bit of Danish. In 2016, he became the CNRS Research Director at the Center for Anthropobiology and Genomics of Toulouse, and he's zooming in from Toulouse today. So now, as you probably know by now, I like to introduce our seminar speakers by mentioning when I first met them, and sometimes we go back to the mist of time, but actually today, I met Orlando 10 minutes ago. So it's really, I'm really very grateful that he would accept an invitation from a complete stranger and also speak to a plant audience. But I'm sure that we can learn a lot about our own problems and our own work by studying this fascinating example of horse domestication. So without further ado, I'll hand over to you, Orlando. Thank you.
Thank you so much for the kind introduction and the kind words, as well as the invitation. I believe that you should see my screen by now, and only the screen and not any of the cameras, right? I guess that's because I have a camera at in the middle of my screen. So if you see the slides correctly, which is okay, then yeah, it was I was very happy to get this invitation, in fact, because for someone working like myself on horses, obviously, you know, Saudi is a place that is of key importance for the origins of the horse and the evolution of the domestic horse, especially the Arabian horse, obviously, right? And also that the invitation came from a plant audience is not too much of a surprise, given that horses are herbivores at the end of the day. So it's no surprise to any of you that our relationship as humans with horses dates back a long time. In fact, horses are the animals that our ancestors have the most represented in the art record of late Paleolithic period, in the form of, for example, this horse figurine that dates back to about 35,000 years ago. It was excavated in Germany, or in the form of course, this beautiful artwork from the walls of the Chauvet cave, also dating back to about 35,000 years ago, this time found in southeast of France, my country, in fact. And for long in our history, our relationship to horses was limited to hunting or art making. But really, this situation changed dramatically, of course, when we first had the idea to domesticate the horse. Because if you think about this process for a minute, you will realize that horses gave us a way to really travel way faster than we could with our own legs, in fact. And that implied that all of a sudden, after domestication, we were given the chance to travel the world in days instead of weeks, or in weeks instead of months, or even years. And with that, that gave us the possibility to bring ourselves, but also our genes, our bugs, our plants, our foods, our culture and languages around the planet in almost no time. So horses really were so much important that I often say that they were the first, they gave us the first opportunity for globalizing the world in history. But at the same time as making the world feel smaller, what horses have given to us is a capacity to make war in a more efficient way and to kill each other in a more efficient way. First in the form of chariotry in the Bronze Age, but then the Iron Age in the form of mounted horseback riding and mounted cavalries. And you see here Alexander the Great to the left defeating Darius III, somewhere around the 4th century before the Common Era. But this doesn't only apply to antiquity and the deep past, because this war making in relation to horse actually lasted to the very 21st century, because this is not less than 15 million of horses that have been actually sacrificed to World War I. So because today horses are no longer used for making war, obviously, but also because the engine motor, motor has actually been developed a lot, horses are almost deprecated, and we don't use horses much more these days, except for some kind of agricultural work as well as for the leisure sport industry. There are about 60 millions of horses living in the planet today, and they are actually spread across 600 more or less breeds around the planet. And all these, I will call them the domestic horses for the rest of the talk, or the modern domestic horses, if you will. And you should picture them, including the Arabian horses, but also the thoroughbred horses, or for example, any type of horse that you can interact with that you would know of in the living on the planet today. In addition to them, there is only one lineage of living horses that is called the Przewalski's horses, that has been considered for long as the last truly wild horse lineage living on the planet. And this is so because they were actually found by the middle of the 19th century, roaming around southwest of Mongolia, northwest of China, and they were actually free-range horses. So from that day onwards, they were considered wild, and they became, in fact, extinct in the wild by the 1960s, but they were saved from extinction due to massive conservation efforts in captivity in zoos, so much so that today there are about 2,000 of them living on the planet. So for people doing evolutionary genomics like myself, you can actually sequence the genomes of the modern domesticated horse that you see to your left and the Przewalski's horse that are living today, and apply some population genetic tools so as to fit an evolutionary model. And the evolutionary model that fits the data that we could generate in living animals tells you that there is a leaky isolation model that is very well fit, in which the two lineage starts to really diverge about 35 to 50,000 years ago, remain in contact for almost 15 to 20,000 years ago, until the last glacial maximum LGM is finished. You realize that the contact was very asymmetric and brought genes from the ancestors of the Przewalski's lineage into the ancestors of the domestic lineage. But from there onwards, those two lineages diverged forever and did not interact with each other much at the genetic level. And the big consequence for us is that if we were to only use a genomic variation as present in modern animals, that means that we cannot leverage this comparison to really understand what took place along the branch leading to the modern domesticate, simply because our outgroup, the Przewalski's lineage, is actually too divergent. It predates the domestication, this divergence, from at least a good 10,000 years. So many of the mutations that will have accumulated along this branch leading to the modern domesticates will actually not relate at all to the process of horse domestication. And so if our objective is really to truly understand the domestication process at the genomic level, then that means that we cannot be satisfied at all with this comparison. It will be simply too noisy. And in fact, it will be even noisier because the model that we can fit actually demonstrates that some genes were introgressed back into the Przewalskis during the captivity in the 20th century. So clearly, using modern variation as preserved in living animals today cannot do a good job in terms of understanding the process of horse domestication at the genomic level. So if we can't be satisfied with the animals that live today, we really have to embark on time traveling. And this is one opportunity that ancient DNA gives us, is the capacity to retrieve small molecules of DNA and deep sequence them so that we could reconstruct the structure and the nature of ancient genomes as time goes, and probably use them to catch evolution red-handed as we were doing the first selections that gave rise to the birth of the domestic horses. And this is those tools that we use in my group and that we have used in my group for over 10 years now, and trying to understand and answer questions such as: when did we domesticate the horse? Where did we domesticate the horse? How did we manage to do so? But of course, given the importance of the horse for human history, we are equally interested in understanding how much domesticating the horse changed patterns of human mobility, as well as warfare and relationships between different civilizations, especially in relation to war. I will not go too much in the details of, how do we sequence those genomes, because I believe that Herman did a pretty good job the last time at explaining that. But just to refresh your mind, of course, you don't do ancient genomics in regular labs. We need very clean clean facilities to be able to not be contaminated by modern DNA roaming around. Of course, our technologies are destructive, but the good news is that we don't need to destroy kilograms of bones to retrieve genomes. In fact, these days, there is enough molecular variation often preserved in 50 milligrams to about 100 milligrams of bone powder, so that we could apply the next generation sequencing platforms and framework by which we extract the whole DNA extract, that is any kind of molecule, DNA molecule that is preserved in a fossil, build them into Illumina DNA libraries, PCR amplifiers libraries, before we can actually plug them in to our most favorite NGS platform, in our case, the Illumina HiSeq or NovaSeq platforms. And even though ancient DNA is very fragmented and the reads are very short, if you sequence deep enough, you can give a chance to your favorite aligners to align back the reads you identify against the reference genome that you like the most, which is, in our case, the third version of the Equus caballus, the horse genome. Okay, so what I'm going to do today, instead of focusing on the methodology, is to try to illustrate the power of those approaches when it comes to understanding horse domestication. And I hope that I will make it very clear that what it is that we have understood that was not even possible to understand after decades of research from the perspective of modern genome variation. So the first idea we had when I started the project, it was about 2012, 2013, was to time travel, but before the domestication times. And the idea was we will actually leverage frozen Siberian remains, as you see here with points 1, 2, 3, because Siberia is very cold. And so we thought, and we were right about that, that the DNA will be extremely well preserved in a sort of natural freezer. And so we found some fossils, 1, 2, 3, dating back to about 4316 and about 5,000 years ago, where the DNA was really well preserved so that we could generate high quality ancient genomes with average genome coverage of about 7 to 24-fold coverage. And because they were dating to 4316 and 5.2 thousand years ago, we simply thought naively that this will give us a chance to see the mutations that the genome, the horse genome, will accumulate from 43 to 16, then from 16 to 5, and from 5 to the modern, 5,000 years ago to the modern time. And that way we could actually recap the sort of movie of mutations coming in the horse genomes as it moved into domestication time. So those 3 genomes were high quality. We had big hopes that we could actually, through them, better understand domestication. And this is the population model that we managed to fit the data to. And in this particular slide and in the rest of the slides that will be coming later, I will show those 3 archaic genomes, ancient ones, as deep blue. And I will always try to represent the Przewalski's horses and domestic horses in green and red, respectively. And so this is the model we had, which was sort of a depressing day to some extent in the lab, because as you realize, those 3 ancient Siberian genomes, they actually cluster with each other, but they are more divergent to any of the living horses today than the Przewalski's and the domestic horse lineages are. To say it in another way, we thought we will actually cover some of these branch at the genomic level, but what we discover is a more divergent outgroup, which was depressing in terms of understanding what genomic changes took place along the branch leading to the modern domesticates. So we had to reinvent the project somehow. What was not depressing, though, is that simply adding 3 ancient horse genomes actually gave us an opportunity to identify a 3rd lineage of horses that no one had any idea even existed. So it shows you to some extent the power of ancient DNA, that even a limited number of samples can actually identify, help retrieve some past lineages of variation that have not made it to the modern world. So I said that we had to really reinvent the whole project. And so what we decided to do instead was to switch gear and to really try to travel at the very first day of horse domestication. To travel to the very first day of horse domestication, we actually considered this site that we call the Botai Neolithic site from north central Kazakhstan, which dates back to about 5 to 5,000 years ago. And the reason why we went there was, as archaeologists have done a lot of excavation in the Kazakh steppe, and this is the Botai site that you see in this slide, where they have actually identified settlements of Botai people. Their houses are buried in the ground, as you see to the left, and they also excavate some pits in which have been accumulated hundreds of bones. I should say, in fact, instead of hundreds of bones, I should say 100,000 of bones, because this is more than 300,000 bones of animals that archaeologists have managed to excavate from the site. And what is interesting to us is that 99% of those bones are horse bones. So really this site, the Botai site, is one really key site when it comes to understand the change in relationship that we had with the horse, because clearly the Botai people developed a new industry, a new subsistence mode, if you will, that was heavily relying on horse exploitation. Also, archaeologists, they have found evidence on site of enclosures, corrals that people actually developed so as to park their horses close to their habitations about 5 to 5,000 years ago. In addition to that, the Botai horses, they show, especially their teeth, and I'm showing you here to the bottom right, a zoom in a premolar tooth of the horse, the 3rd one, and you can see some sort of V-shaped structures at the surface of the tooth, which is a typical damage pattern that horse teeth actually develop when they are bridled. So we also know that the bowtie people bridled their horses and probably were using them for horseback riding. But in addition to that, the bowtie people were also milking their horses. We know that from isotopic signatures retrieved from fatty acids that have been preserved at the surface of their vesselry. So clearly the bowtie people, they were probably domesticating the horses already, and they were probably the first to do so. So that's why we could reasonably hypothesize that by sequencing the genome of the bowtie horses dating back to 5,000 years ago to 5.5 thousand years ago, we could actually catch evolution red-handed at the very first time when we domesticate the horse for the first time. And so we will retrieve the sequence variation present in the ancestral genome, the one that we first domesticate. So this is the data set that we generated a few years ago. We sequenced about 25 ancient bowtie horse genome to relatively good coverage. And to put that in perspective, we also sequenced about 100 other horse genomes spread across Eurasia that I show here as DOM in orange. That are actually dating back to the last 4,000 years to sort of follow the genomic development post-bowtie domestication in the genome of the horse. And to be honest, this is the model that we could fit to the data, and that came as a big surprise to us, because as you can see, the bowtie horse genomes were actually placed on this branch, leading not to any of the modern domesticates still shown in red, but actually straight leading into the Przewalski's horses, the only horse that was considered until that work as the last truly wild horse living on the planet. And those two branches, they did not exchange much genes because this first lineage only contributed to about 2.7% of the genome variation present in the modern domestic horse lineage. So to summarize what we find, even though we were looking for the ancestors of the horses that have been domesticated, what we find at the genomic level is that the bowtie horses were first domesticated by some people, probably in Central Asia before 5,000 years ago. But this domestication process did not give rise to the modern domesticates that we know of today. In fact, it gave rise to the only horse that was considered as wild. So that means that this first domestication process actually was deprecated, became sort of abortive to some extent. So the very important implication is that it must be that some other people somewhere else have managed for a second time to domesticate the horse. And this is the second domestication process that actually gave rise to the range of genetic variation present in the modern world today. So we know that this shifts from the first domestication process to the second domestication process took place between 5,000 years ago to 4.2 thousand years ago as the latest and the earliest radiocarbon date estimate that we have from the fossils underlying the genomes that we have sequenced. So we know, and for the rest of the talk, of course, I'm going to talk about this second domestication process as the DOM2 process, if you will, to save time. So that answers the when question. The horse domestication took place at least twice, and the second one actually took place about 5 to 4.2 thousand years ago. We know precisely the time range, but it doesn't answer the where question if it's not at bowtie that this thing took place. So for the last 3-4 years, what my lab has been focusing on is trying to find an answer to the where question. And what we decided on doing is actually sequencing many more horse genomes from across Eurasia, as you can see here, ending up publishing a few months ago 264 additional ancient horse genomes spread across Eurasia. And the y-axis here, the vertical one is actually reflecting time in thousands of years ago here. And you see that we really did a strong effort to actually document the horse domestication, the horse genome variation around the time that I was referring to, around 5,000 to 4,000 years ago, but also to place these findings in longer term perspective, we also sequence horse genome from across Eurasia up to more than 50,000 years ago. And so this is the kind of genomic variation that we could identify in this gigantic genome data set, which is represented here as an MDS plot, a multi-dimensional scaling plot, in which the two axes that you see here and there actually reflect the genetic proximity. So 2 points will be closer to each other on the graph if they share a larger fraction of the genomic variation, and the vertical axis here reflects time still in 1,000 years ago. So in that particular plot, what you see is things that I've already commented at, the Przewalski's horses, the Dom2 horses are there, or the Botai horses that we focused on, also those archaic horses that I mentioned before as well. But you see that we uncovered quite a range of variation that we did not have any idea even existed. The light blues, the purples, and the brownish, the reds, and so forth. So clearly you see that by accumulating more ancient genome sequences from across Eurasia, we keep on discovering forms, genetic forms of lineages that we had no idea existed. So it seems that the picture became a lot more complex, but luckily for us, this could be actually organized in a very simple manner. And this manner was following some geographic trends. So if I project on a Eurasian map, the genomic ancestries that we found in the different lineages roaming around Eurasia over before 5,000 years ago. So here, the larger the pie chart, the greater the number of ancient horse genomes that we sequence at a given location. And the different colors, they represent the average ancestry components that we could identify in the genome. What should strike you here is that you see some colors distributed according to geographic locations. You have the light blues to the left, you have the deep blue to the east, you have here the green in the Central Asia, and you have the orange in this part of the world. It seemed to us that really the genomic evidence was structured according to phylogeographic patterns, really. You didn't have the same genomic variants, if you will, depending on where you were living across Eurasia, if it was prior to 5,000 years ago. And so given this evidence, we could actually leverage patterns of dissimilarity through space to actually infer, using the EAMS software, those migration surfaces that you see here in dark brown if there is no migration, or in very dark blue if there is tons of migration. And so what it uncovered is indeed a Eurasian world that was without any migrations in the horse world before 5,000 years ago, which explains that how those different lineages could actually form according to geography. The situation did not change much when it comes to 5,000 years ago to 4.2 thousand years ago. And again, you see a lot of evidence of no gene flow, dark brownish isolation surfaces across Eurasia. But really what struck us is that from 4.2 thousand years ago onwards, the genomic distribution of horses really changed dramatically, because all of a sudden, only one genomic component, which is represented here in orange, started to expand across Eurasia and became absolutely dominant at the scale of the whole continent, demonstrated that horses indeed were actually not local anymore, but became global, and that the world had no barriers of migrations anymore. So this is the time 4.2 indeed, where the second domestication process took place and really had a very rapid success, because in a few centuries, really, the same horse type actually expanded across Eurasia as a whole, but also replaced all the previously existing lineages that we uncovered. This is also at that time, between 4,000 and 5,000 years ago, that we could identify in our data a massive expansion of the maternal lineage and a massive expansion on the paternal lineage. that really demonstrate that the past breeders, they really managed to control the horse reproduction like they never managed to do before. Those plots are Bayesian skyline plots that we can identify leveraging patterns of mitochondrial variation through time to the left, or patterns of Y chromosomal variation through time to your right, which indeed indicates that the effective size of the horse population, both for paternal lineages to the right and maternal lineages to the left, were multiplied tenfold during that millennia. So clearly, not only we control the reproduction of horses for the first time to scales that were unrepresented, but we leverage these scales to actually supply the now global demand of horses that those civilizations needed at that time. So because we had such a strong geographic phylogeographic pattern in the horse genomic data prior to domestication, prior to DOM2 domestication, I should say, what we tried on doing is to use these patterns to train deep neural networks so as to predict what was the origin, the geographic origins of any of the horses that we have sequenced that will be younger than the last 4,000 years, including down to the modern world. And so we can take one genome dating back to 3,000 years ago, another one dating back to 3,000 years ago, and use the deep neural network to ask where were these ancestors living at the surface of the planet. And we did this exercise for over 200 ancient horse genomes dating back to the last 3,000 to 4,000 years. And regardless of the time considered that they lived today or 1,000 years ago as diamonds, or as squares 2,000 years ago, or even more as circles, we had the very same pseudo-likelihood projections that told us that this is where the DOM2 horses all originated from, regardless of where and when they have been identified and sequenced in our data sets. So this pseudo-likelihood surface actually uncovers the genetic homeland of the DOM2 horses. It answers thus the where question. We know now that we managed to domesticate the horse for the second time, 4.2 thousand years ago, north of the Caucasian Range, northwest of the Caspian Sea. And this is from that very tiny place that all the horses that have been used and domesticated since originated from. So we were left with the how question. How did we manage to do so, to succeed in domesticating the horse for the second time? And what were the biological reasons, if any, for such a massive expansion success? So the idea we had to answer this question was to really consider horses as if they were of two types. Those that were successfully expanded across the planet, the DOM2 horses on one hand, and those that never managed to do so. And when you have two groups, what you can easily do is FST scan the whole genome to try to find regions of the genomes, maximizing the shift in allelic frequencies between those two groups, which we actually did at the scale of what you see here, the 31 pairs of autosomes of the horse genomes. And really what we had as a result was really unanticipated. We had two strong, outstanding regions, one in chromosome 3 and one in chromosome 9, as you see here, both of which neared fixation in DOM2s versus the non-DOM2 horses. So if I zoom in these two positions of the genomes, starting with chromosome 3 here, the ZFPM1 locus that you see here. Basically, this is a region of the genome that we can actually manipulate with some drugs, especially baclofen, and changing the expression of that gene, ZFPM1, actually ends up in modifying the behavior of mice, of male mice in the lab. And the most important change that you can actually trigger that way by, again, modifying the expression of the ZFPM1 gene is to change the aggressivity behavior. Now, if you overexpress the ZFPM1, you actually end up having a lot more aggressive male mice in the lab, and you can measure that quantitatively because they will bite at each other a lot more. So because this gene is so much key in terms of controlling some aggressivity-like behavior or anxiety-like behavior, it is, we think, reasonable to hypothesize that these very first locus that is heavily selected in the DOM2 lineage from the very first day of DOM2 domestication was probably selecting from for a sort of more tamed behavior. And maybe these DOM2 horses had the success they had simply for being able to develop an easier behavior to interact with the early herders. So that speaks for one of the two loci. How about the second one? So the second one, just to refresh your mind, is in chromosome 9, and this time is just upstream within the promoter of one gene, which is called GSDMC, and these GMDSC genes when it's overexpressed in some humans, it is associated with a chronic syndrome that is called the spinal lumbar stenosis syndrome. And to make a long story short, this syndrome ends up changing the size of the spinal cord that you see here. And because the vertebrae would then press onto the spinal cord much more than in sort of wild-type persons, it ends up making the walk extremely painful in humans. Both in this association has been uncovered both in Asia and Europe. So we think again that this is reasonable to believe or to hypothesize that this horse variant that has been selected really to almost fixation in the DOM2 horses was a variant that was actually making the locomotion, the walk less painful. So when I'm asked what it took to successfully domesticate the DOM2 horses, I'm really tempted to say two things: a more tamed behavior to some extent, and also a stronger horseback or a back that made the walk a lot less painful. What is really interesting, at least in my opinion, is that at the same time as we see the DOM2 lineage expanding across Eurasia about 4,000 years ago, we also see an expansion of equestrian equipment in archaeological material in the form of those horse bits, for example, that you see here, spreading across Eurasia hand in hand or together with the DOM2 horses. So this is in the form of those horse bits, but this is also in the form of, especially in Asia, of chariots as well. So the Sintashta culture that is represented here was dating back to 4.1 to 3.8 thousand years ago. And this is world famous for the invention of the spoked wheels that you see here. Here you see them transversal, and that would be a chariot and the human buried with the chariot. And you could probably see two horse skulls here and there. So this is a typical grave or burial for the Sintashta culture. So we think that this archaeological evidence, together with the demographic expansion that we saw, plus the geographic expansion that we uncovered, really shows that this is a key timing for horse domestication. And indeed, horses, but horse equipments, chariots, were actually fueling at that stage human migrations, at least in Asia. What is really interesting is that the same work that we've done on horses, other labs, for example, in my old labs in Copenhagen at the Center for Geogenetics, but as well as in Harvard University at the David Reich's lab, they have uncovered for years now genomic signatures of human expansion in Asia at about the same time, 3.7 thousand years ago, 4,000 years ago. And they uncovered this because they saw that some genomic ancestry, which is represented here in deep blue, that was originated from this region that could play as the horse domestication center north of the Caucasian range, actually started to expand in Asia at about the same time as horses. 3.7 thousand years ago, we recover this ancestry originating from the steppes here into almost Mongolia and then kicking in East Asia. So early in Asia, this population of humans actually leveraged the horse as a new locomotory force to really sustain large-scale migration, bringing into Asia new language families such as the Indo-Iranian language family. So clearly here, this is a key example of horses and human evolution coming hand in hand. What we show, though, for Europe is something different, because this step-like ancestry that I'm showing in the blue in human genomes only expands already into Europe 4.7 thousand years ago. So at least a good half millennium prior to the domestication. So we have here in Europe a different situation than in Asia, in which the human populations are spreading across the Indo-European language families into Europe some 4.7 thousand years ago, actually were not fueled by horses in opposition to what we saw in Asia. So, of course, for the last 4,000 years, the horse evolution and selection and domestication didn't stop. And we could also, leveraging our data, to actually try to unfold this process of us selecting and engendering new forms of horses in the last 4,000 years. We could do that because we have full genome data. So we could actually take any single location of the genome and reconstruct patterns of expansion of any variant through space and time. So I will only illustrate that today for the sake of time for a number of characters that we've been interested in, first of which the size variation, asking the question, can we see evidence in our data about when is it that we started selecting horses of different sizes across the world? And this is a collaborative work that we've just published a few months ago, again, in collaboration with my really good colleague, Professor Jin Yang from Beijing University, in which we uncovered a variant of the TBX3 gene located on chromosome 8, that it's a single nucleotide variant, a G or A, that is associated to about 20% of the size variation that we see today in Asian horses. And we could actually demonstrate using KO mice that we could drive the overexpression of the TBX3 genes, and that will be associated causative for longer size of the limbs at different stages of the mice development. So we had a driver, a genetic allele that is a driver of larger sizes in horses. So we could actually go back to our data, all the genomes we had across Eurasia, and use them to actually plot the frequency changes of the G allele, the driver for larger sizes, across time. So time is the x-axis, y is the allelic frequency, and we were really struck to see that this unimodal increase of the G allele through time is really a signature of positive selection. It kept on being selected for larger sizes, and this process started about 2.3 thousands of years ago, which is clearly the time when the first dynasty emerged in China, the Qin dynasty. And this is a dynasty that is extremely well popular for the terracotta army that is world famous, really. So this is about the time when breeders in Asia started to select horses for larger sizes. And the same story goes in Europe. We have other variants that are actually driving size variation into European lines, such as the Z-fat variant, one particular variant at a location of chromosome 9 of the horse genome that also started to unimodally increase in frequency at about the same time, about 2.5 thousand years ago. So when it comes to size and larger sizes, really the early breeders were not so much into larger sizes, and this is only in the last 2.5 thousand years that they were starting to be interested in developing larger sizes of animals. So about the coat coloration, for example, we could do the same exercise for example, the chestnut coloration. And we see here a picture that is very different, in which breeders were first really fancied a lot, this cut coloration, but then it was deprecated in the Middle Ages before it came back to fashion or to become attractive again during the most recent times, the last few hundred years. We could do the same for speed at a short distance, leveraging the variation at different chromosomes, including the PDK4, as well as the AC and 9 variants that are actually associated with better performance at flat racing at short distance. And what we found was that these alleles only rose in frequency very late during horse evolution, late being the last 1,000 years. So when it comes to speed, I guess, this is only a very recent creation that we have some very speedy horses at short distance because past breeders before 1,000 years ago were not really selecting for such characters. So the same exercise, you can easily understand that we could do that for any single trait that we know the genetic determinism of, so that we could also apply those temporal patterns at the genome-wide scale diversity. So this is what I'm showing you here, in which time is still the x-axis, and to the left graph, what we calculate is the heterozygosity level of every single genome of ours. Each point is a genome, and we calculate heterozygosity as a measure of diversity and effective size of the population, where you see that for a really long time after Dom2 domestication, after 4.2 thousand years ago, the past breeders, they managed to really sustain a highly diverse horse population. But the situation changed dramatically in the last 200 years because we detect a drop of heterozygosity in the range of, on average, 16%. So it seems to us that for 4,000 years, horse herders managed to save a lot of the genetic diversity, but the last 200 years had a lot more impact than the 4,000 years before in terms of losing diversity. And as they lost diversity at the genomic level, horses, they also were enriched, those genomes, in deleterious variants that you can see to the graph to your right, which is the genetic load, which is the total sum of deleterious variants that we could detect in the genome at homozygous sites. So as conclusions, I hope that I've convinced you that ancient genomes can indeed be very informative for understanding complex processes such as the domestication process. And in terms of take-home messages, we've seen that there are only 2 lineages of horses that still live on the planet today, but they have been all the descendants of 2 independent domestication processes. So when it comes to say, is there any wild horse still living on the planet? I'm afraid the answer is a big no. There is none anymore, simply because the Przewalski's horse are the descendants of the first domesticated horses and must be described then as feral instead of truly wild, in fact. The Dom2 lineage expanded from the Pontic-Caspian steppes around 4.2 thousand years ago, and then and onwards, it gave rise to all the domestic horses that exist and have existed since on the planet, and replacing, therefore, all the previous populations of horses that existed. And of course, during the last 4,000 years, further selection and modern breeding practices reshaped the horse genome. The last thing that I want to show you is the extent of the change. Again, this is the MDS plot that I've shown already, where this is all the genomic variation that we have uncovered using ancient DNA, of which the modern variation only represents that range. So in terms of diversity loss in the last few thousand years that have took place in the horse population, it has been gigantic. And this is one of the key aspects of ancient DNA, to give us the ability to travel back in time and uncover those genomic alleles or this genetic variation that has left no descent in the modern world. And with that, I would like to thank you all again for the invitation, the attention you had today, I guess. And also, I would like to thank all my sponsors, including the ERC, for supporting this kind of research. And with that, I will be very happy to take any questions if you have any.
Thank you very much. That was a real tour de force of horse domestication, ancient DNA, population genetics. I would say real entertainment value also, in addition to, of course, the excellent science. I can see there's a virtual applause going on. So normally we start our questions with questions from the students and the postdocs to try and encourage discussion. So any students and postdocs who wish to, or early career scientists who wish to raise a question. Elodie.
Yes, hello, a very nice talk, Ludovic. I have a question. You say that recently we have lost diversity in for the horses, and I was curious which lineages are disappearing. I mean, what's less represented nowadays, and will it change?
That's a very fair and very good question. So the drop that I mentioned is average, so it's about 16% average and genome-wide, right? But if, of course, if you think, and I should have said, but this is related to the last 200 years, which is concomitant with us doing breeding, meaning inbreeding, reproducing the horses that look the same so as to fix specific phenotypes to some particular breeds, I guess. And you know this in plants more than what we do in animals, I guess. So this very process of population fragmentation actually was the driver of the loss of diversity. And in fact, of course, when I say that, you realize that those breeds that have been demographically the most affected by the drop, so those breeds where the founder effect event was larger, I'm thinking thoroughbreds, for example, but there are other breeds that have been actually created by really only a handful of founders, they are the most affected. And in some of the breeds, it can go to even like further drops of diversity up to 40%. And the bad thing is that it's not just a drop. A drop is bad in terms of long-term evolutionary potential for a population, I guess, but this is also that at the same time that you lose on average, you enrich the deleterious variants. And this is simply because as you reduce the population size, as you therefore increase the drift, you actually reduce the potential of negative selection to filter out of the negative variants from the population. And therefore, the same process not only results in impoverishment of the diversity, but an enrichment of the deleterious variants, which requires thus serious measures for conservation of the lineages. Even though the census size of those lineages looks great, you know, you have thousands of horses of a given type, their genomic health is actually a lot more endangered.
Any other students or postdocs or early career scientists, let's say, before we go to the group leaders and professors? Come on, guys. In that case, Rod, I can see you've got your camera on. You may go ahead. Oh, hang on. Alice?
Hello, Ludovic. Thank you very much for the presentation. It was fantastic. And continuing on this line, yes, of course, deleterious mutations are a consequence of the domestication process. As you decrease the genetic diversity and you raise the frequency of this mutation. So is there any way, is in any way possible to reintroduce some diversity in these horses by utilizing those that are like the only one left that are like kind of the feral, as you said, of the actual domesticated horses to increase diversity, to unload the charge of deleterious mutations?
Yeah, that's an excellent suggestion. In fact, I mean, we are advocating to make use of genomic tools really to inform on which population should be prioritized in terms of those to be used as a source population to actually, inject some additional variation into some of the lineages. But the problem we have is that the populations that we will try to save that way would be those that are actually called breeds. And so oftentimes what people want to keep is their breeds, because this is, the heritage of centuries of selections, or also breeds are often associated to the identity of a particular terroir or a particular region or a particular cultural identity, I guess, right? So it's not an easy process. It's not like genomic tools could theoretically save everything somehow, but they will come to clash with some local cultural and ideological decisions to make, and which are equally important to consider, right? So that would be one way. Another way that people have started to envision will be to really leverage or harness, if I may say so, the power of CRISPR-Cas9 technologies and also cloning in horses, because clones, even though they are identical genetically, you can use the same animal, the one with lower genetic load, if you will, as a sort of breeder for the next generation, as opposed to just select the best stallion, the one running the fastest, for example, right? So you can imagine that you identify those animals that are the least loaded, and you use them to create each generation more crosses, more matings with different mares. So this is something that has been actually discussed. And also, if you were to not only clone, but CRISPR-Cas9 them, then you could probably knock out a few of the deleterious variants. And so in a few generations, you could probably do something good. But again, this second thing will be actually clashing a lot more at the cultural level, especially in Europe, I guess. And there are only a few handful places in the world, such as Southern America, where cloning is legal, where these things will be allowed in, I guess.
Yes, indeed, this is also like the case for wine in Europe, where genome editing is not allowed, and also traditionally it's some cultural heritage that we have. I'm Italian, so yes, we have culture behind, and you are French, so
I understand.
Yes,
I understand.
Yes, completely. So yes, thank you very much.
Okay, Rod.
I really enjoyed your talk. I had I have a bunch of questions. So hopefully I can get through a couple. So can you cross the Zawaski horses with modern horses? And is the are those horses viable?
Yeah, they are. This is, again, a very good question. So I didn't even have to do it. It has happened in zoos oftentimes and time again. In fact, we demonstrated that at many levels, but it didn't take genomics to demonstrate that, because the Przewalski's horse karyotype and the domestic horse karyotypes are not the same. There is an extra pair in the Przewalski's. So if you take a Przewalski mom and a domestic dad, basically, you will have a F1 hybrid that is fertile and non-sterile with 2N69 instead of 2N64, 2N66, right? So clearly we know this is possible. And as I said, this is even fertile. So then you can backcross that thing with modern domesticates, or with Przewalski's, or even with hybrids. And I must say that then it took the genomic tools to really assess how often this took place. And we were very surprised that even though there is a very documented pedigree in the Przewalski's, because they spent the last 100 years in zoos almost, we noticed many mistakes in this pedigree, and we identified many instances of what you just described taking place in zoos, because they place them in the same enclosures, I guess, or you use some substitute surrogate mothers to actually milk the Przewalski's horses, and this took place a lot. So our genomic estimates were that on average, it depends on which pedigree of the Przewalski's you look into, but the worst was nearing 17% of introgression of domestic blood lines into the Przewalski lineage. So short answer, yes, and it has taken place a lot, in fact, in the last 100 years.
Yeah, I was just curious if that would be a way of introducing more genetic variation into modern horses, and maybe that could be interesting. So the two genes that you identified for anxiety and back pain, I'm curious, have people looked at those genes in camels?
I'm not aware of that. So
Because they've been, domesticated, and is their behavior and stuff. I think that would be kind of a cool thing.
absolutely, I totally agree. It doesn't ring my mind. Like, I don't remember to have seen any genomic paper on camels addressing those two genes. The literature that we managed to track was in lab models such as mice, rats, and guinea pigs, but also depending on which of the two genes, but also the medical literature in humans, like big time. But no, and clearly this is something to investigate, two candidates to look into camels. Also,
it'd be easy to do, but it'd be kind of interesting to look at that. So thank you very much. I'll turn it over to the next questioner.
Right, so we've got Mark, then Haneen, and then we'll go to the chat box. So Mark.
Actually, the two questions in the chat boxes are actually very similar to what I was going to ask. So why don't you let Robert and Saleh
yeah, please, Robert.
And I'll just add a supplementary to each of them. Thank you.
Robert, do you want to read out your question, or shall I do it? Do you want to come online, Robert?
Sure, no worries. Thanks first of all for the talk. It was super interesting. So I was wondering whether you found more of your candidate adaptations in behavioral patterns, or was it more selection for physical or physiological adaptations? So basically, is running faster more important, or not biting the owner?
So yeah, exactly. So thanks. It makes a lot of sense. And this was actually one of the incentives for us starting this project, really to assess and quantify which traits, biological ones, were, super important to unlock, to make even domestication possible, right? What was locking domestication from the animal perspective. So depends on the time period. From the early days of domestication, we have those two massive candidates that we have been able to see. There is a third one that I didn't mention, because the interpretation of that thing was a lot less straightforward, because it seems to be a master gene for reprogramming methylation, DNA methylation. And so, the downstream targets can be actually genome-wide. And so we really had to make more studies to really make sense out of that. The second thing that I should say is that when it comes to early domestication times, those that we spotted are very easy to spot, simply because there are single nucleotide polymorphisms or blocks of the genome, haplotypes, if you will, that will be positively selected and uniformly selected. But when it comes to other traits, they could have like minor contributions of to any phenotype, and you could have like hundreds of them contributing, if not thousands. And so they are pretty pooled. Our sample set is poorly designed to have really some power to detect those. So I will really refrain to make very strong conclusions about those traits were more important than the other ones. What I think we should say from our data is that amongst those traits easy to find, those that are sort of Mendelian inheritance and driving most of the phenotypes with a few variants, then it seems that behavior and the horseback was key. But when it comes to even behavioral changes, I'm sure that we are not even scratching the surface of the iceberg here, especially in horses, where the variance of behavior, it takes 20 years to really quantitatively study in stalls and such. So the short answer is we don't know. When it comes to now, not the early days, but the latest changes during domestication, say the last 500 years, here I can say confidently, yes, that aesthetic changes as well as speed has been key, because these are the traits that have been heavily studied by the horse community. Probably they are those associated with, US dollars value, basically. So clearly people have looked into that very carefully.
Yes, myostatin and so on. Actually, could I ask the supplementary on that then?
Sure.
Just about behavior. I've always wondered about dogs and dog genetics because there's so much incredible breeding for behavior specifically. So I love Robert's question. Actually, his question was more subtle than mine. But I was just wondering if you're able to transfer any. I don't know the dog literature, so maybe you could give us a briefing on that, because I think that'd be pretty interesting, given that this brain, maybe other people's brain, is so amazingly fantastic, and the genetics of that still is so obscure, at least as I understand it. So yeah, can we learn anything from dog genetics, and then can you transfer that sideways to horses?
Yeah. So I'm going to try, and it's not an easy one, and thanks for the question. It's very interesting. Obviously, I'm very much advocating for such studies to not be restricted to horses, but to be actually developed in dogs, in cattle, in sheep, goats, and camels. Why not? Obviously. And in fact, there are groups around the world. I'm thinking for dogs, Gregor Larson at Oxford University has been doing that for years now. They are a bit lagging behind in terms of number of genomes, but they are really catching up very fast. So as I'm speaking, I cannot really be very precise in the cross-model comparison for the lack of data in the case of the dogs. But what I can tell you is that the LD, in dogs, is actually much stronger than in horses. So it means that this sort of, probably the last 200 years have been more impactful in the dogs than I've even said in the horse, which speaks even to be more worrisome in the case of the dogs than in the case of the horse, because of course the variation has a long range effect at the scale of the genome. So that diversity is a lot less important, I guess. And in terms of behavior, I don't recall from the published papers that they had evidence of such genes as those that we saw. But what they found, though, recently is evidence for potentially 2 domestication processes in dogs as well. This is still debated, but there is literature about that. And it strikes me to say that this is what we uncovered for horses as well. So shall we view this as a sort of rule or an exception, that there is more than one single domestication locus? For the 2 models that seems to be the most studied with ancient DNA, horses and dogs, it seems that it's more the rule now than the exception, surprisingly. And the second thing is that when it comes to size, we have this single SNP polymorphism that is associated with size through time, and they discovered something similar in dogs very recently, but where the large size, if I recall correctly, was actually the ancestral size. So it will be the other direction around, like where some wolves were larger and then were selected for smaller sizes, and then back again to larger sizes much more recently in horse domestication. So we would love to do such comparisons, and that's why I should have said that perhaps, but we developed at CNRS an international framework that we call Animal Farm for doing so, and that involves Gregor Larson's from Oxford University working on dogs, but also colleagues in Trinity College working on cattle and sheep and goats.
Yeah. Thanks. Brande, you've got a heap of questions flooding in.
It's amazing. You've stimulated really good discussion.
Exactly.
But I'm going to try and take them in the order that they appeared now. So I think the next one is Sauli, or Saul. Yeah. I'm not sure how to pronounce it. And then we'll go to you, Hanin. Sauli, could you...
Yeah, thank you so much for the talk, Professor. I have a bit of a general question out of curiosity. Have you looked at the origin of the Arabian horse? And I'm particularly curious if it was actually brought from like Turkmenistan, from their horses, because I'm actually from Kazakhstan, and we know the horses like of Turkmenistan, the Halteke breed. And when I came here in Saudi, I actually thought that the Arabian one looks very similar to the Turkmen Halteke one, except for the face. So I'm a bit curious if you have looked at it.
And can I just jump in? And just ask?
Sure.
On some of your figures, you had some shading coming Transcaucasus across into the Transcaucasia. region. And I'm just wondering if you can make a commentary on tranquil cases, which is very related, I think, to Saule's question.
Yeah, so this is very good questions. And you said, Saule, that you were curious, but you're not only curious, but your intuition is correct. Actually, genomically, the Akaltiki's horse genome is actually very close and actually branching off from the Arabian diversity early. So they are very related indeed. I haven't looked specifically yet in the form of published papers at this very question, but I can tell you that other groups have, based on modern day variation, and we have, in fact, but not published that, looked at that question using ancient horse genomes. It seems that the Arabian horse is quite diverse still today. You know, we picture, at least in Western Europe, the Arabian horse as, you know, uniform splendor. Really, it's a beautiful animal, but really very uniform and probably also associated with some genetic disorders that are very, very, very famous. The people like Samantha Brooks in the US at the University of Florida has actually genome surveyed populations from across Egypt to actually Iran and the peninsula, obviously, and she uncovered about 5 to 10 times more variation that was expected from the perspective of those Arabian horse lineages that are heavily selected, those that are selected for endurance racing, really, right? So good news is that it seems that they are a lot more diverse than what we thought. The other thing that I can tell you is that from our unpublished work, we really do see them becoming massively influential from about the 7th to the 9th century CE, which is absolutely not surprising given that it's coincidental with the Islamic expansion, obviously. And we see their genomic influence to be massively important, especially first around the coast of the Mediterranean range, but then onwards, becoming global. I guess the Arabian horse, I think it's safe to say that this is not because you're in Saudi that I'm saying that. This is very, very true. Has been probably, with thoroughbreds, the most influential breed in the last few centuries. And thoroughbred, as you know, is actually partly originating from Arabian horses. The Dali Arabian horse is one of the 3 founders. So to summarize, Akaltikis and Arabian horses, yes, very much related indeed to Arabian horses, very a lot more diverse than was expected, which is great as well. And third, they have been probably the most influential breed living on the planet. And 95% of the horse bloodlines that exist today have Arabian horse blood.
Thank you.
Thank you. Let's continue. Haneen. We can't hear you, Haneen. We still can't hear you, Haneen. Okay, so while Haneen sorts out the technical problem, let's go back to the chat box. There's lots of questions there.
Okay.
Oh, okay, Haneen.
Yeah, at some point also I lost you, sorry. Okay, now I can hear you and you can hear me. So thank you so much for the question. I have maybe simple question. So I know in Kazakhstan, they're actually eating horses. So I'm not sure if in your population, you actually have selected horses based on the use, like horses for sport or horses for eating. But if you have, then is there any differences in the genetic or the genomic variation between the two, the horses that people eat and the horses people use for race or other use? And if yes, then if there is actually any selection that actually made these people to use these horses to eat, but not for any other use. Because I also think that they don't actually mix both. There are just for race, for example, and specific horses to eat.
Yeah, you're very right, Haneen. So basically, I know for a fact that they eat horse meat in Kazakhstan because I've been flying there a few times, and every single time working on horses, I'm served some horse meat. So clearly, I can actually testify that this is correct. You have to remember that in Western Europe, especially and including in my country, but this is also true and even truer, in fact, for the UK. There's been a Christian ban on horse meat, dating back to a pope living in the 7th century CE, if I remember correctly. And from that time onwards, actually, eating horse meat has been associated with negative perception. So clearly in Western Europe. So clearly this is the meat that surprisingly people will not go to unless they are really starving, right? When you starve, you eat whatever is available, but other than that, you don't. Surprisingly, in France, there are still some butchers preparing the horse meat despite these sort of cultural and religious barriers, right? So, and because of that, because most of the industry has been driven by breeds coming from Western Europe, eventually, the thoroughbreds, specifically the standard breds, are descent from British horses and so forth. There has not been so much work done on trying to identify those loci driving larger muscular sizes in horses. Those genes that we know of that make the horse meat larger, so make an animal more productive, are really located, such as the myostatin gene, are really identified in the context of flat racing. Because for being a sprinter, you need massive muscles at some particular parts of the horse anatomy. So they have been actually, surprisingly, with regards to your questions, they have been identified, surprisingly, in relation to speed racing and not with horse meat eating, basically. Also, in terms of market, I'm not sure it will represent quite an incentive as opposed to speed. So the trait that has been the most studied thus far has been in relation to speed and flat racing.
Yeah, thank you.
That actually relates to one of the questions in the chat box. Amin, is there anything you'd like to add to that?
No, thank you. The question was the same, actually. So yeah, I think
I will confess I was also thinking of that question. Okay, and so no more to add to that, Amin, is that right? Or
no, thank you.
Okay, there's two more questions in the chat box. One is from Asil. You still with us, Asil?
Yes. So I think thank you for the talk. I just had a general question, and if you could explain how the influence of politics and economics and the trajectory of between humans and the evolution of horses, if you have any idea about that.
Yeah, so thanks for the question. I love this question so much so that, when it came to the time when I applied to ERC, I had the choice to go to biological science or to what they call the human path, so humanities. And I chose humanities, even though I'm a molecular biologist, for answering this question, because I think this is where the novelty lies, really, to try to unfold history from the perspective of the animal and the animal technologies. So how come animal or plant technologies, in fact, become political instruments? What is the advantage that those things give to power at one time? And maybe this advantage actually drives expansion of civilization or drive the capacity of one winning the wars over the other, right? So this is, in general framework, this question makes total sense to me. And I think that this is through such questions that we could really reconcile humanities and quantitative genetics, which are two worlds that have not communicated so much thus far. So I can actually give a few examples now, and some of the things then that we are trying to unfold as I'm speaking. So two examples that I think are becoming very clear from our patterns, from the data that we have already generated and published, is this Indo-European language family expansion, or the Indo-Iranian language family expansion, which is massive. Indo-European language and Indo-Iranian languages across the world, it's 70% of all the languages that have ever been spoken, right? English is one, German is one, Danish is one, and so on and so forth. So those language families, without the horses, they may not have got such a success, right? Of course, there is the colonization in the last 500 years, but even beyond that, as I saw 4,000 years ago, it was not Britain, Spain, and France invading the world. It was other kind of civilization expanding on a horseback and bringing those languages across the planet, across Eurasia, in fact. So one is they gave a massive political agenda to those moving along with the horses. This language became a sort of lingua franca, if you want, a sort of international language that actually spread across the world. So that's the first one. The second one, we alluded to that, that's the Arabian influence. Clearly, this new horse type that the Arabian people invented somewhere around before the Muslim expansion, but kicking into Europe during the Muslim expansion, became a sort of new fashion. And clearly, if it didn't give a political advantage, it certainly gave an economical advantage, because it became so popular. So, I mean, clearly it must have explained some of the economical trajectory that those societies took place. And then what we are trying to do now is to take some periods of the past that are associated with massive economical or political shifts and see whether the horse can explain some of the observations that they have in humanities. And I'm thinking the Roman times, for example. When you have the Roman times in Europe, they expand big time. In Northern Africa, they expand up to almost the Black Sea, and they expand even in the UK, so pretty much everywhere. And they have a very, the logistics of such an empire is very, very important until they collapse in the 4th to 5th century CE. And what we're trying to see is whether horses or donkeys or mules have played a role in the logistics and expansion of these empires. And what we already published actually a few months ago was that prior to the Roman times, you have only horses almost in Europe and some donkeys, but at tiny, tiny levels. But during Roman times, there is a horse type that was not around before, which is the mule. So the F1 sterile hybrid between a donkey and the horse. And this became really, really important. And there are some sites where 50% of the animals, of the equine assemblages that we find are actually mules. So, and this is coming hand in hand with the Roman expansion, because mules are known to be, more resistant. You need a lot less water to breed them. You need a lot less food to keep them. But of course, they are a lot more expensive to produce because they are sterile. So you need both parents, and you can't breed them generation after generation. You have to rebreed them at every single generation. And despite the economical costs, Romans have developed them big time. And once the Roman Empire collapses, they disappear. Donkeys are there and horses are there, but no mules anymore. So we really think that this is an evidence for, when you have an empire that lives of the capacity to trade at long distance, to bring tools to the army thousands of kilometers away, clearly developing mules has been one way to make Roman Empire possible. But when the economical incentive disappeared, when actually the empire started to collapse, 4th, 5th century CE, mules collapsed as well and disappeared from Western Europe. So clearly you see that more than the horse, but the equine resources, horses, donkeys, and their hybrids alike, can actually be used to really explain how the different empires from the past have taken some of their power and economical wealth, I guess.
Thank you. We've got one more question in the chat box from Maria Navarrete. You want to join us, Maria?
Hi. Thank you very much for the talk. It was great. I was just wondering if it's possible that the populations of free-roaming horses, like the ones that are in the US, that they are not actually like true wild horses, but horses that were domesticated and now are free-roaming, that these populations could recover the genetic diversity similar to the ones that were in the ancient, really true wild horses. And is it going to be like a process of de-domestication in these populations?
So your question is spot on. I mean, we are finalizing one manuscript as I'm speaking about that very question, and actually the different questions that are related to that. So I'm going to try to focus here. So first, what are those free-roaming populations that live still in the US? You think about the Mustangs, right? And if you picture the American West, these are the horses you think about, obviously, right? So the mainstream narrative, and our data backs up that, is that actually they are not the descendants of wild horses that were once roaming in Northern America, but actually they are all the descendants of horses that came together with the colonization, the conquests, right? So they are sort of Spanish/British horses that actually went back feral. And then, when you have something like the South American West, I mean, you can only expand if you are a horse, right? So they are clearly feral and not truly wild from that perspective. The second thing is that, of course, since this process took place there at different points in time, you can actually think about this as a sort of natural experiment in which you have at different point back in time, different fertilization processes starting. And so now that you could go in the field and sample and genome sequence those horses, you can ask the question, what is the amount of deleterious variants that are still present in a population that only came back feral, say 10 generations ago, or 20 generations ago, or 30 generations ago, or 50 generations ago, and try to measure quantitatively the time it takes generation-wise to actually become truly feral. Like, when is it that you reverse back to a sort of wild state? And then the question that you could ask is domestication even reversible? Which is, of course, a question that is quite of an interesting one ever since Darwin days, right? This is one of the question that he had in the book about animal domestication, about domestication and speciation. So the short answer to that is not simple, though, but for sure we measured situations in which the load was still very high in some feral populations, and because the demography was very small, in fact. So it's a trade-off between demography and non-direct exposure to human activities, in fact, that matters in terms of how fast you go back to equilibrium, if it makes sense.
Okay.
Yeah, thank you very much.
I'm a little bit conscious of time, although oh, sorry, someone's calling me now. I'm very conscious of time, even though the discussion has been very, very good. I think we should probably draw it to a close, unless anyone has any burning issues.
Can I ask one very short technical question? Did you focus all your analyses on single nucleotide polymorphisms, or can you also technically study structure variation?
Yeah, very good one. So for now, of course, this is on our agenda to look at structural variation. For now, you have to realize that the average size of every single DNA fragment that we sequence is about 50 base pair long. And so in terms of having high resolution of even paralogs or, like multi-copy genes locally in the genome is really hard. So we have some, say, genetic changes that are small-scale deletions, like 11 base pairs. Those ones, you can picture how you could actually study them easily. But when it comes to 100 base pairs long, structural variation and more, it's hard to do at the moment still. This being said, we generated the data in a way, and I should have said that perhaps, that my group has invented a number of methods by which you can leverage the sequence information, but also predict DNA methylation and nucleosomal positioning in the past. So we are very interested, as I'm speaking, into exploring those two sources of information along the gradient of course domestication. And what we hope we're going to find is some patterns about systematic shifts of DNA methylation and nucleosomal repositioning in relation to using horses with mounted warfare or even like horseback riding. For this, we leverage the fact that ancient DNA is damaged. Oftentimes, the fact that this is damaged is a pain in the bum because it's damaged, you can't sequence as much as you want, but here this is an advantage because the way this is damaged is actually a direct linear function of protection for the nucleosomes or a direct function of, deamination of citizens leads to urasols, but it leads to thymines if you're methylated, and our sequences can be used to see this difference. So that means that you can say at the sequence level, at the single nucleotide resolution, whether or not a CpG is methylated. And so, of course, the same way we have those allelic trajectories through time, we would love to see very soon epi-allele trajectories through time to sort of see, even hotspots of epigenetic variation, which can maybe just tell you something about shifts in environmental exposure. So there's definitely lots to do. And to finish answering your question, of course, those two things, I'm relating them in the same questions because they are related. Transposable elements can expand and invent the genome as a function of methylation of the genome. So probably this is a way we could find and leverage to also study structural variation eventually.
Very exciting. Thanks.
Right. So before we completely exhaust, I've got confused if it's Ludovic or Orlando because
Orlando is my family name, but I mean I go by both. It's fine, no worries.
So before we completely exhaust you, I think we should bring it to a close. And thank you very, very much indeed for not only a stimulating seminar, but a very extensive and stimulating discussion. So thank you very much.
Thank you, Fabian.
Please don't completely leave because I just wish for a few more minutes to have a little chat with you.